Okay. I would like to thank the organizing committee for the invitation to this conference. So the precise mechanism of most chemical cardiomyopathies remain elusive despite decades of research. And as you'll know, a major hurdle is the lack of appropriate model due to the inaccessibility of human cardiac tissue. Animal models do not accurately recapitulate the disease phenotype because of interspecies differences, and current models do not allow to predict an adverse drug event for an individual patient. In my talk today, I will talk about human pluripotent stem cell technology that might provide a solution to this modeling problem. I will talk about the possibility to use this technology for cardiotoxicity in a dish, and maybe to apply it for predicting chemotherapy-induced cardiomyopathy. And finally, I'll talk about our current study with in vivo modeling of human diseases, what we call next-generation disease modeling. So it all started with the first type of pluripotent stem cells, the human embryonic stem cells. These cells are derived from the inner cell mass of the blastocyst, and they can be propagated indefinitely in their undifferentiated forms. They were generated more than 15 years ago in our institute. The cells can be differentiated into derivatives of all three germ layers, including cardiomyocytes, and Yitzhak Ehad from our group generated the first human cardiomyocytes from these cells. And throughout the years, we have used this technology to establish models for drug screening for QT prolongation and conduction abnormality. And we have shown in a series of proof of concept studies that these cells can be used for regeneration of the infarcted myocardium. In this example, in rat model of chronic infarct. But human embryonic stem cells have several limitations. We are unable to generate isogenic patient and disease-specific pluripotent stem cell lines using this technology. We have a limited set of human embryonic stem cell lines. So these cells are non-autologous for the rest of the human race. And of course, we have major ethical barriers. And the potential solution to this problem came with the introduction of the human pluripotent stem cell technology introduced by Shinya Yamanaka. Briefly, this technology is based on reprogramming adult somatic cells with a set of transcription factor into pluripotent stem cells called induced pluripotent stem cells, IPS shortly. And these cells are very similar to human embryonic stem cells, and they can be differentiated into all derivatives of all three germ layers, including cardiomyocytes. And then we can use these cells for maybe in the future for regeneration and for drug screening and toxicity screening. And shortly after the introduction of the IPS technology, we have been able to generate cardiomyocytes from human IPS cells, and we have shown that these cardiomyocytes are very similar to human embryonic stem cells. We've also been able to generate high-throughput drug screening systems using genetically encoded voltage indicators, what we call JEVIs, and you can see here the fluorescent signal actually giving us an indication for the voltage in the membrane, and we can measure the action potential duration, which reflects the QT interval in the ECG. So you can know that non-cardiac drugs such as cisapride, erythromycin, and even the tyrosine kinase inhibitor, nilotinib, cause significant action potential duration prolongation, actually what reflects QT prolongation. So we can use this technology, the pluripotent stem cell technology, to have to ask questions about safety and safety of drugs and toxicity of drugs. We can tailor a specific drug to a specific therapy to individualize therapies. We can use this as disease in dish models for target identification and to identify novel therapeutic targets. And finally, we can use them for patient-specific autologous cell therapies for regenerative medicine purposes. But the next question is whether we can use this system for modeling chemotherapy-induced cardiomyopathy. And as you all know, and I will start with anthracycline cardiomyopathy, there are several mechanisms involved in anthracycline-induced cardiotoxicity, which include reactive oxygen species, impaired mitochondrial function, abnormal calcium handling, and inhibition of the cardiac isotype of topoisomerase 2B, all lead to cardiac cell death. And recently, a group from the FDA showed nicely that doxorubicin induced dose-dependent cell apoptosis in human pluripotent stem cells cardiomyocytes. So you can see the reduction in cell viability, the dose-dependent reduction in cell viability, and the increase in DNA fragmentation. But interestingly, what they found out with this system is that doxorubicin actually upregulates death receptors such as the FAS, the TNF-related apoptosis-induced ligand receptors DR4 and DR5, and TNF receptor 1. So if we add TRAIL, the TNF receptor ligand, even the TRAIL application actually enhances the cardiotoxic effects of doxorubicin in human pluripotent stem cell cardiomyocytes. And you can see here the fold change after TRAIL application. So actually what happens and what they found out is that doxorubicin upregulate death receptors such as the TRAIL receptor, the DR4 and DR5, and circulating death ligands interacting with these receptors cause apoptosis and tissue damage and then mediate cardiotoxicity. These death receptors may serve as a novel therapeutic markers, novel therapeutic targets, but also as an early marker for the disease. When we zoom in into the molecular level, doxorubicin application induced upregulation of gene clusters associated with mitochondrial damage and Ross metabolic pathway in the human pluripotent stem cell platform. And importantly, in this study they used pathway analysis, they revealed a similar transcriptome profiling to that of dilated cardiomyopathy and hypertrophic cardiomyopathy. This group also applied the lessons of cardioxan protection mechanism, and they knocked out using the CRISPR-Cas gene editing system the TOP2B, the topoisomerase isotype of cardiac cells. And by doing that, they actually rescued the cells from DOX damage. And you can note here the increase in cell viability and decrease in DNA fragmentation. However, the knockout of topoisomerase 2B did not affect cardiac differentiation of the human pluripotent stem cells. The human pluripotent stem cells model can be also used for high throughput screening, and this is one example from a group from Stanford, a very active group in the field. They used a panel of tyrosine kinase inhibitor and applied it on human pluripotent stem cell cardiomyocytes, endothelial cells, and fibroblasts, and assessed the cells in multiple ways for gene expression analysis, for contractility, calcium imaging, and electrophysiology, for quantitative toxicology and phosphorylation arrays. And you can see the nice panel of tyrosine kinase inhibitors and the automated cell viability assessment, and they found out 3 tyrosine kinase inhibitors have the highest effect on cell viability. These are sorafenib, regorafenib, and pontanib. And these 3 tyrosine kinase inhibitors are actually inhibiting the VEGF receptors and the PDGF-B receptors. And when they looked into pathway signals, the VEGF and PDGF receptors inhibiting tyrosine kinase inhibitors cause dose-dependent alteration in tyrosine kinase signaling. And you can note here the downregulation of VEGF and PDGF signaling, but no effect on RB4 or EGFR, but a compensatory augmentation in insulin and IGF signaling. So the hypothesis is that tyrosine kinase inhibitors actually downregulate the AKT, the anti-apoptotic pathway, and there is a compensatory increase in insulin and IGF signaling, and this may be a potential therapeutic target when we might add insulin or IGF to rescue the cells from the cardiotoxic effects of these cells. When they assessed for electrophysiological effects of tyrosine kinase inhibitors, you can know that both nilotinib and vandetanib cause action potential duration prolongation. As I noted earlier, this reflects the QT interval in the ECG. But Gleevec or other tyrosine kinase inhibitors did not prolong the action potential duration. And taking together all these parameters, they generated a cardiac safety index for tyrosine kinase inhibitors. And this cardiac safety index nicely correlated with reported clinical toxicities and FDA black box warnings, as you can note here. So we have shown that the human pluripotent stem cell platform can be used to model chemotherapy-induced cardiomyopathy, but the next question in the era of personalized medicine is whether we can predict the effect of chemotherapy on an individual patient. And this study also came from this Stanford group. They generated human pluripotent stem cells from breast cancer patients. There were 4 breast cancer patients that, after 1 year follow-up, did not develop doxorubicin-induced cardiomyopathy. They tagged this group as DOX. 4 breast cancer patients that developed doxorubicin-induced cardiomyopathy. This was tagged as DOX-TOX. And 4 healthy controls. And they assessed, using multiple parameters, the doxorubicin damage. They assessed contractility, calcium cycling, metabolism, gene expression, apoptosis, and chemoprotection. And as you can note here, the human pluripotent stem cell platform was able to identify the effect of doxorubicin on multiple parameters, such as cell viability, reduced in the DOX-TOX group, those that developed the chemotherapy-induced cardiomyopathy, comparing to the DOX group, the patients that did not develop the chemotherapy-induced cardiomyopathy, DNA damage, abnormal calcium handling with prolonged tau in the DOX-TOX group, and oxidative stress, here evaluated by mitochondrial superoxide and ROS levels. When they zoomed into the molecular level, they identified that in the DOX-TOX group, there was downregulation of biological processes such as muscle contraction and cholesterol biosynthesis, and upregulation of programmed cell death and P53 downstream pathways. So using this technology, they actually identified the vulnerability of the patients that developed chemotherapy-induced cardiomyopathy in more than a dozen mechanisms of doxorubicin toxicity. These are marked in blue here. But this model is not without limitations, and the human pluripotent stem cell model failed to identify the protective effect of cardioxan, as you can note here in DOX-TOX and in DOX patients, and was able to identify a protective role for N-acetylcysteine. So this does not correlate with what we know from the clinic. There are additional several limitations of the human pluripotent stem cell in vitro model. These cells are relatively immature and may not accurately reflect the disease pathogenesis in adult individuals. Using these models, the cells they lack 3D spatial organization, and we evaluate only cardiomyocytes. So we do not evaluate the multicellular composition of the cardiac tissue, including endothelial cells and cardiac fibroblasts. We are unable to simulate the adult metabolic energetics and the modulation incurred by the immune system. And we are unable to assess drug pharmacokinetics, which is so important for assessing chemotherapy-induced cardiomyopathy. What we are currently doing in the lab, especially for genetic cardiomyopathies, but also for chemotherapy-induced cardiomyopathy, is using in vivo modeling of human diseases, what we call, using IPS systems, what we call next generation disease modeling. And the concept is very simple. We take human pluripotent stem cells and differentiate them into cardiomyocytes. We can inject them to a skid mice, and then we get very nice human islands, human cardiac islands within the mouse heart. Or we can differentiate them using an engineered tissue platform and mix them with endothelial cells, fibroblasts, and collagen, and transplant these beating calamaris, this engineered tissue, to the mice. And we use this technique for modeling a very challenging disease to model, arrhythmogenic right ventricular cardiomyopathy. This is one of the most arrhythmogenic syndromes and a common cause for sudden cardiac death in young athletes. The hallmark of the disease is lipid droplet accumulation. And when we try to model this disease in vitro, this is an adult onset disease, we didn't succeed to get lipid droplet accumulation, and we got it only by applying very aggressive metabolic stress. So actually applying very aggressive metabolic stress masked the pathogenesis of the disease, and we had problems with this model. And we used the in vivo model to model this disease. We got very nice stable grafts up to 3 months of human cardiomyocytes within the mice heart. And you can note here that the human cardiomyocytes differentiated very nicely. They were much more differentiated than the in vitro cells. And when we assessed for lipid droplet accumulation, you can see here the ARVC cardiomyocytes and the control cardiomyocytes. Actually, the in vivo environment promoted the cardiomyocyte maturation and uncovered the disease phenotypes. And you can note here the positive immunostatin for perlipin, the marker of lipid droplet accumulation. So we can use this model to model ARVC without an additional metabolic stress. And we are currently using this in vivo modeling technique to model chemotherapy-induced cardiomyopathy, and we will be very happy to collaborate on that. So in summary, pluripotent stem cell technology is a unique platform for assessing the effect of chemotherapy on human cardiomyocytes. Disease mechanistic insights, drug toxicity screening, and identification of novel therapeutic targets may be achieved by using the human pluripotent stem cell-based platforms. The human pluripotent stem cell models may be able to identify patient-specific vulnerability to chemotherapies. And finally, in vivo modeling may improve the accuracy and applicability of this technology in the future. I would like to thank you for listening, and I will be happy to have questions.
Thank you very much. We have time for a couple of questions for the first 2 lecturers before we move to the clinical topics.
How long does it take to do this
On the in vitro model or the in vivo model? For the in vitro model, it's very simple. So we have beating cardiomyocytes with genetically encoded voltage indicators, so we can assess their electrophysiological parameters in 2 hours. For the in vivo models, we need 1 month. So we need to inject the cells, wait 1 month until the cells mature, and then apply the chemotherapy. So this is a very good question, because I'm not sure that using the in vitro model we can assess the magnitude of the damage. But we hope that using the in vivo model, this is one thing that we assess now, we will be able to correlate the dosage to the intensity of the damage and to assess the magnitude of the damage. Assess